Enzymes are biological catalysts which basically act to speed up the reactions and processes that take place within living organisms, for example, within the human body. Now, enzymes have a very high specificity, and what that basically means is enzymes react with a specific type of molecule or with a group of molecules that are closely related. Now, the molecules or molecule that reacts with our enzyme is known as the substrate, and the substrate binds to a specific location on the enzyme known as the active site. And the interaction between our enzyme and the substrate is electric in nature. The force involved is electric in nature. Now, when the enzyme binds with our substrate, this complex is known as the enzyme-substrate complex. So in most cases, for the enzyme to actually function effectively and efficiently, another type of molecule, a non-protein molecule, has to bind to our enzyme, and this molecule is known as a cofactor. So cofactors are non-protein molecules that basically optimize the activity of our enzyme, and cofactors can be broken down into two categories. So we have our metal ions, which are basically inorganic cofactors, and we have the coenzymes, which are the organic cofactors, and the coenzymes are usually vitamin derivatives. So we have two main types of coenzymes. So coenzymes can be further subdivided into these two groups. We have the co-substrates, which are basically coenzymes that bind to our enzyme via loosely held bonds, while our prosthetic group is basically the coenzyme that binds to the enzyme via tightly held bonds, for example, covalent bonds. So let's discuss in more detail what the difference between these cofactors are. So metal ions include the minerals such as magnesium, zinc, and potassium, and so forth, while our coenzymes are basically vitamin derivatives, and this includes biotin and thiamine pyrophosphate. Now, biotin is basically a water-soluble B vitamin, specifically it's the B7 vitamin, while our thiamine pyrophosphate is also a B vitamin derivative. Now, our coenzymes can be subdivided into these two groups. But what exactly is the major difference between these two groups as discussed earlier? So we have the prosthetic group, which basically bind tightly to the enzyme. So these coenzymes bind tightly, usually via covalent bonds, to our enzyme and remain tightly bound during the reaction. And during the reaction, they are not actually changed. And so following the reaction, those coenzymes are regenerated and they remain unchanged. However, co-substrates bind to our enzyme via weak bonds, and usually the co-substrates end up transferring one of the groups on that co-substrate, on that coenzyme, to our substrate. So following our enzymatic reaction, these coenzymes, known as co-substrates, usually change themselves, and to actually regenerate that co-substrate, we have to undergo some other type of reaction that usually involves energy. Now, one example of a prosthetic group, basically a coenzyme that tightly binds to our enzyme, is the heme group. And we'll talk more about the heme group when we discuss hemoglobin. Hemoglobin is basically a biomolecule or protein that binds oxygen and carries oxygen to the different tissues found in our body. Now, an example of a loosely held group that we're going to discuss is our coenzyme A, which is basically a molecule that is involved in cellular respiration. Now, an enzyme that is without its cofactor is known as the apoenzyme, while the enzyme that has the cofactor bound to that enzyme is known as the holoenzyme. Now, the question still remains: how exactly does the substrate actually bind to the active side of our enzyme? So we have two different models that basically describe two different ways by which our substrate can bind to our enzyme. We have the lock and key model as well as the induced fit model. Now, the induced fit model is the theory that basically is accepted nowadays, while the lock and key model is the older model that is basically no longer really accepted, and we'll see why that's the case in just a moment. So let's begin with the lock and key model. This model presumes that the enzyme's active side is a perfect keyhole for our substrate, which is basically the key. So it states that the active side is a perfect fit for the substrate, and neither the substrate nor the enzyme actually changes shape during our binding. So this is basically the diagram that describes the lock and key. So we can basically imagine that the enzyme has this active side that is a perfect fit for this substrate, in the same way that our lock is a perfect fit for our key. And when we place that key into our lock, the key nor the lock actually changes in shape. They do not change in shape, and so this is our lock and key model. So the substrate shown in red fits perfectly inside the active side of the enzyme, and no change in structure actually takes place. So this is our lock and key model. Now, the more accepted model, what we think actually takes place on the molecular level between the substrate is the enzyme and the enzyme, is described by the induced fit model or the induced fit theory. So this is the much more accepted model. It states that the active side is not exactly a perfect fit. However, as the substrate basically goes into the active side, what happens is the active side, the actual enzyme's active side, as well as the substrate itself, actually change in shape ever so slightly to basically create that perfect fit. And that makes sense because if we examine the subatomic level, if we examine the molecular level, and we discuss the electric forces involved, whenever we have the interaction between different molecules, these electric attractive and repulsive forces will basically change the shape of the molecule just a little bit, and this can be basically explained from a physics perspective. So basically we can imagine our enzyme initially has a rectangular active side, while our substrate is, let's say, some type of trapezoid. Now, when the trapezoid, the substrate shown in red, actually goes into this rectangular active side, the rectangular active side changes in shape slightly, and the trapezoid, we can imagine the trapezoid also changing in shape. For example, it is basically lengthened. So we see that the substrate as well as our enzyme are not really perfect fits, but when they interact within the active side, the enzyme as well as the substrate, they change in shape to basically create that perfect fit. And this model is known as the induced fit model, where the lock and key model basically states that the active side shape and our substrate shape does not actually change in shape in any way because they are essentially a perfect fit. So this fits perfectly into the active side of the enzyme. So this theory doesn't really hold very much when you think about it from a physics perspective, because we know from physics when our molecules interact because of the electrostatic forces, electromagnetic forces, whenever molecules are essentially approach one another, there has to be some change in shape of those molecules as a result of those electric forces. And so the induced fit model is the model that is basically the accepted model nowadays. It describes the way that our substrate interacts with our enzyme.